I'm honored to introduce our next speaker, Dr. Brandon Fields, co-founder and CSO at Fieldstone Bio. He has a deep domain expertise at the leading synthetic biology lab at MIT, led by Christopher Voigt. His keynote's going to be on synthetic biology, hyperspectral imaging, and AI, advanced environmental monitoring for agriculture, climate, and national security. Brandon, you can go off mute and take it away.
All right. Thanks for that introduction, Evan. So today I'm going to be talking about how we're using living intelligence for environmental surveillance. Just a bit about us and how we came together. I'm a synthetic biologist from MIT, where I worked under Dr. Chris Voigt, one of the world leaders in this field. We partnered with Patrick Stone, who has a 20-year career in government, leading technical programs for national security. And together, we're translating this technology from the lab at MIT to solve some real-world problems. So when we think about environmental monitoring, it's really low throughput, right? Manual methods. Think of farmers out in fields collecting soil samples to measure nutrients or pathogens. Environmental remediation experts or the warfighter trying to identify where harmful chemicals are before they harm us, right? In all these situations, the techniques are very accurate and specific, but they're really low resolution, right? You only get data at that exact spot that you actually measure. So going back to the farm example, if you have a 2,000-acre farm and you have a handful of data points, you're just trying to extract out useful information from a limited data set. Now, remote sensing solves this scale problem, right? Currently, we have hyperspectral imaging on drones, planes, and satellites, and we're generating tremendous amounts of spectral information about the environment. And we can tease out information from this spectra to make decisions. But currently, we're really limited to chemicals with really large footprints or easily identifiable signatures. Things like methane gas leaks or the greenness of the leaves on these farms. Right? But there's a whole world of chemicals that are in this data. They're just really hard to see. And we use biology to solve this problem. And so I love showing this picture because it really gets at the scale and power of biology. Every single corn plant you can see in this picture has over 1 trillion cells. Okay, and these cells are constantly sensing and responding to information to better the plant. Right? And there's a whole nother world of microorganisms that cover nearly every surface of our planet. And these microbes have evolved to sense and respond to chemicals in their environment. Now, as synthetic biologists, we can isolate these microbes and engineer them in the lab to sense chemicals that we're interested in. But one of the major bottlenecks in this field and why it really hasn't been adopted is how do you visualize this activity? How do you visualize these tiny microscopic sensors from really far away? And that's what our technology bridges. So what we can do is engineer microbes to sense chemicals and turn on a hyperspectral reporter, right? So this is something in the cell that changes its spectral properties into a designable format for us to easily tease out from hyperspectral imaging data. So we can apply the engineered microbes to the environment where they can sense the chemical of interest, turn on one of these reporters, and then using our hyperspectral imaging data and AI pattern recognition, we can identify where in the environment that the microbe actually sensed the chemical. So really, the microbe is translating the presence of a chemical of interest that's generally hard to see in the data into an easily identifiable format. And just to show you how this looks, all right, so here's one of our microbes that we've isolated from soil in our labs, and then we've engineered it. We've changed its DNA to carry a program. And what this program does is senses arsenic and turns on one of these reporters. On the right, you can see this rocky, sandy soil where we've contaminated it with arsenic at different concentrations. We then applied our microbial sensor at equal parts throughout this whole image. Okay, and what you can see is that by eye, you can't tell where the arsenic is, right? In fact, you can't see the cells, even though there's trillions of them in this one image. Right? And if you want to know where the arsenic was, current methods, you would grab a physical sample and send it out for analysis or average all of the soil here and get one value. But with the hyperspectral imaging and AI, we can identify where the microbe is actually sensing the arsenic and creating our reporter. Right? So we're not only seeing where it's sensing arsenic above a certain threshold of arsenic levels, but how strong that signal is, right? Meaning there's more arsenic. And so it's with this technology that we're able to actually get spatial information about different and unique chemicals in the environment. With old technologies, you'd have to take thousands of soil samples to get this resolution. And so just to end, I've talked about how we're using microbes to sense chemicals. But synthetic biology is fundamentally changing our world, right? This whole concept of removing microbes from the environment, manipulating them, and then replacing them. When people think of synthetic biology, we typically think of health-related things like personalized disease interventions or engineered probiotics, right? But there's also this environmental aspect, nitrogen fixation on fields, bioremediation or biomining, new materials like living apparel, living paints, and self-healing concrete, and even further out there, things like terraforming planets or ocean deacidification, right? All based on our ability to take the power of biology to solve key challenges for our planet. And I'd like to thank everyone for the time and the opportunity to chat today. Thanks.